Hello, today we are going to talk about Anchor 6 genetics. If we look at the first slide, let's learn and review some vocabulary terms that are needed for Anchor 6. The first vocabulary word is allele. You can think of allele as a different version of a gene. This would be, example, like a factor, allele, trait. They all mean the same thing. If we look at the other vocabulary words to remember, it is genotype and phenotype. If we look at the word genotype, we see gene in that word. When we look at the genotype, we're looking at the set of alleles or genes that an individual has. A genotype is represented by a pair of letters, like capital B, like capital R, little r, or like little b. So anytime you see letters, they are referring to the genotype. If we look at the word phenotype, phenotype starts with PH, and that represents the physical appearance of an organism's genotype. In other words, we judge people every day on their phenotype. We say they have blonde hair, we say they have blue eyes, we say they are tall, we say they are short. So when we look at somebody, we are looking at their physical appearance or their phenotype. Two other important vocabulary terms dealing with genetics is homozygous and heterozygous. The difference between the two is homo means the same. It's the condition of having two of the same alleles for a gene, like capital B, or lowercase b, lowercase b. They are the same letter. Heterozygous, hetero means different. And you can look at the T in it like two different letters, like a capital letter and a lowercase letter. Capital B, little b. Capital R, little r. If we look at the next slide, that is using a punnett square to predict the outcome of the cross between a pea plant that is heterozygous for yellow seed color and another pea plant that is heterozygous for seed color. So if we look at this punnett square and we do it, they're heterozygous for yellow. We would have a capital Y and we'd have a lowercase y. These are the genotypes. And another heterozygous, we'd have a capital representing dominant and we'd have a lowercase representing recessive. And we go to use this punnett square, we would do one from the father, one trait from the mother, which they will be both capital. One comes down from the father, one comes over from the mother, one from the father, and you always put the capital letters first because it's dominant. One from the mother, or vice versa. One from the mother, one from the father, and we look at this punnett square. And if we look at the punnett square and we tried to make our predictions, our genotypic predictions would be 3 to 1. There would be 1 capital Y. It's hard to write letters like this. Ratio is written with the colon. 2 capital Y, little y, which is heterozygous, and 1 homozygous recessive, little y. That's the genotypic ratio. The phenotypic ratio, again, a ratio is written with a colon, would be 3 yellow and 1 green, which would be the recessive. Okay, so now we go on to the next slide. Types of inheritance patterns. You have complete dominance, where one is dominant, one is recessive. In other words, you either have the trait or you don't have the trait. Freckles or no freckles. Widow's peak, no widow's peak. It's black or white. Another type of inheritance is polygenic trait. That is a trait influenced by several genes, where one gene does not determine the trait. Like height is made up of many, eye color is made up of many, skin color is made up of many. The third one is incomplete dominance, where neither allele is dominant. You have hair texture, so you would have straight, wavy, or curly. You would have an in the between, an incomplete dominance. And then you have multiple alleles, is when there are 3 or more alleles in a population. And then you have co-dominance, co meaning it shares the dominance, like AB blood type, where there is not one that is more dominant than the other. If we look at this slide here, it's dealing with pedigrees, where the males are squares and the females are circles. When it is shaded in, that means that that individual, whether it is male or female, has the trait. If you look at this over here, the autosomal, if both males and females are shaded in, then we say it's an autosomal trait. If on the bottom over here to your right, the sex link, if you only see one sex shaded in, like here you see only males that have it, we refer to it as a sex-linked trait or a trait linked to their sex. A review of Anchor 6 Part 2 is gene expression. With gene expression, the central dogma, it's the entire process by which proteins are made based on information in the DNA. So if you could recall, we had DNA is transcribed into RNA. RNA leaves the nucleus, and that is why it is transcribed from the DNA, because RNA can leave the nucleus where DNA is trapped. The RNA then, once it leaves the nucleus, goes to the ribosome where it is translated. And if we can think back to Anchor 1 Part 2, the cell organelle ribosome, its job is protein synthesis, which means makes a protein. So if we look over here at the picture, transcription means the copying of DNA into RNA. The RNA leaves the nucleus and goes to the ribosome, where it is translated 3 bases at a time, called a codon, into an amino acid. Amino acids form a protein. So when we look at the reveal of the steps of gene expression, otherwise also known as aka protein synthesis, we look at transcription. Transcription, DNA is copied into RNA. It is complementary to the DNA. Remember, C pairs with G and A pairs with U, and we know it is RNA because the base is U instead of the DNA base T. Translation, RNA is translated into a protein. The protein is made up of amino acids. If we look at this slide, mutations. A mutation is a change or a variation in the DNA. A point mutation is when you just change one base. But if you change just one base, that can cause different types of mutations. One being a substitution, where one base is switched with another. It can be silent, which means it is not affected at all. It's the same amino acid. It could be a missense, where it is told to change the amino acid from one valine to another serine. Or it can be a nonsense, where it's told to stop before it should. Another example of a point mutation is an insertion, where you insert one base where it is not needed. Or you delete one base where it is not needed. DNA technology involves the application of genetics for practical purposes. Pretty much, you can know that genetic engineering is all man-made. It is technology, and it involves man manipulating the DNA. We have restriction enzymes. Restriction enzymes, basically, if you look on the picture here, they cut. They cut the DNA into smaller, specific sequences. And when they restrict it here, cloning with plasmids, you have the restriction enzyme, and you put it in the donor, and you get a recombinant DNA, which is recombined with the new. So it's the old plus the new, what you want, and then you make copies of it. And this is a way that they have manufactured human insulin, as well as other genes. Recombinant, you recombine the DNA for what you wanted, and you make copies. Also involved in genetic engineering, DNA fingerprinting. We talked about this, the gel electrophoresis, where you cut up the person's DNA with enzymes and you run it. And if the banding patterns match, they show that they are genetically related. The smaller pieces move faster and they get to the end quicker because they move faster. The larger pieces are slower and they remain at the top. We use this to determine paternity for crime scenes as who did the crime. You can do it to identify a victim that has been in an accident and you burned severely and you can't identify them by looking at it. That concludes the review for Anchor 6.